Okay. So Adi, if you see that we are not recording, but it seems that we are recording.
Yes, because it's automatically done.
Okay, good. So, hello, welcome. Thank you for being with us today in this workshop where we hope to discuss with you about the opportunities to transition from in vivo to in vitro for tetanus, diphtheria, and pertussis containing vaccines. This is an AFSA collaboration workshop. Today, we are actually going to discuss specifically on the opportunity for the safety testing and just a few rules for today. So this is a webinar. So the attendees will be able to interact with us through the Slido that you can see here. So you can get to the Slido code through the QR code, or you go to the slido.com and type the code that you can see here. We are going to go through the presentations with all the speakers that we have, and then please do write your comments into the Slido. We will try to bring them up if we see that they are brief during the just after the meeting, the presentation, or otherwise we are going to discuss it during the discussion time that we have for today. So it would be nice also to understand who you are. So when you type your comment or your question, if you can also write who you are, because for us it's going to be important to understand from where the question comes from. So without further ado, I would like actually to give you some additional information about this workshop. So probably it's not the first time that you are joining our workshop. So this is part of the Humane Society International project that is focused in accelerated non-animal batch release testing for human vaccines. Within this project, we are communicating with a lot of stakeholders in order to understand what is their needs to implement those non-animal testing, understand what kind of complexity they might encounter, and try to work with them in supporting their implementation of an updated strategy. We would like also to thank a lot all the members of the project International Steering Committee that are supporting the creation of the agenda of this and all the other workshops that we created so far. So for today, again, we are going to give you some information about the available opportunities. So what has been developed and what is still in the development in terms of the safety testing opportunities. We would like to hear from you about the knowledge that you have about those opportunities. If you had some experience on the introduction of these or the challenges that you encountered while considering the introduction of this type of methodologies. What is important for us is about understanding what is the best strategy, and we know that every organization, being a company or a regulatory agency or national control laboratory, might have a different perspective or might have different needs or strategy in order to do so. We are here today to understand exactly how those type of strategy can be developed and put into place, having the concrete implementation of those ones. So very briefly, this is the agenda for today. We are actually going to get into the details about all these opportunities, and please stay with us for the interactive discussion, because we really want to have the feedback from you. And again, don't forget to connect to Slido so we can check your questions and your comments during the entire event. So that's it from my side for now, and now I'm happy to give the floor to the first speaker.
Okay. The first speaker for today is Catherine Milne. Catherine works with DTwP European Directorate for Quality of Medicine, the Directorate of the Council of Europe. Within DTwP, Catherine is the head of DBO, of the Department for Biological Standardization. Catherine, we are very happy to listen to your talk.
Thank you very much, Heidi, and thank you for the promotion. In fact, I'm head of Networks Division here in the Intergovernmental Committee and Networks Department, where Laurent Mallet is the head of department. And I'm happy to talk to you today about issues around safety testing for diphtheria, tetanus, acellular pertussis, toxoid vaccines. So as noted, I work at EDQM, and there we go. And so I'm going to talk to you a little bit today about the requirements for toxicity testing of these vaccines and the work that the EDQM has done in implementing alternatives to animal testing. So EDQM, as you probably know, is part of the Council of Europe, and we have many activities in the field of the quality of medicines and healthcare. One of the activities that you're probably familiar with is the activity of the European Pharmacopoeia, which creates reference standards and records methods as documentary standards for the quality of control of medicines. Works through the European Pharmacopoeia Commission, which is a treaty-based body, and the Biological Standardization Program Steering Committee oversees the development of biological reference preparations for this work. All of the work of the European Pharmacopoeia is embedded in the principles of the European Convention for the Protection of Vertebrate Animals, used for experimental and other scientific purposes, which is in fact the precursor to the European Directive 86/609, which is now replaced by the Directive 2010/63, and which we also follow. So the primary areas through our activity at EDQM are seen here on this slide. We have first the Pharmacopoeia, which I've already mentioned. This has common quality standards that are applicable to human and veterinary medicines. Importantly, these texts are legally binding on the same date to all convention signatories, and this includes the EU, 39 signatories plus the EU, and 30 observers. All of the texts are reviewed regularly to identify 3R opportunities, and you'll see later how this has impacted the area we're going to speak about today. There's also the Biological Standardization Program. This was established in 1994 and is co-sponsored by the Council of Europe and the European Commission. The program runs large-scale collaborative studies to establish reference standards and methods for biologicals, both for human and veterinary use, with the goal to include them in the Pharmacopoeia. We also work on method verification for implementation and inclusion in the Pharmacopoeia. And international harmonization is also a key goal because we know that medicines are global commodities. The third area where we have heavy activity in the 3Rs is in the Official Medicines Control Laboratory Batch Release Network, both for human and vet. And here the OMCLs are involved in applying the 3R mechanisms for batch release for human and vet vaccines, using the fewest animals possible, including implementation of in vitro methods where possible. And they're also some of the key developers for the 3R methods and contribute heavily to the collaborative studies in the Biological Standardization Program. And if you're interested in more information, there's some links to our websites there. So the European Pharmacopoeia, forever in the introduction, allows for the use of alternative methods. This is a standard procedure, but it was recognized at some point that sometimes this wasn't being done, in particular for the 3R methods, because of issues not with the new methods, but of issues with the existing in vivo methods. So the group 15 and 15V came up with a new chapter, 5.2.14, called the substitution of in vivo methods for the QC of vaccines. And then somewhere in the text, it's and potentially other biologicals. So it's known that the introduction of the in vitro methods to replace in vivo methods was often presented because of the characteristics of the in vivo method themselves. These were methods that had been used historically and shown their value, but they had problems with the variability. The validation status was not at the same level as the common validation criteria that we expect now with respect to the ICH principles. And the product attributes assessed were sometimes assessed differently or were in fact different product attributes altogether. So the demonstration of equivalence in this context was not only problematic, but also in many cases of limited relevance. So Chapter 5.2.14 provides guidance on how to introduce alternative in vitro methods where a head-to-head comparison with the in vivo method is not relevant. And it envisages possibility that the validity of the in vitro method be demonstrated without such a comparison, with the concept of substitution as an alternative approach for a direct head-to-head replacement. The focus throughout the whole chapter is on scientific rationale behind the in vitro methods and relative to what is provided in the current in vivo methods. So in this presentation, we're going to focus on the safety and the absence of toxicity in toxoid vaccines. I'm going to start with diphtheria, tetanus, and acellular pertussis. These are vaccine components produced from active toxins that are deactivated during the production process, so going from a toxin to a toxoid, which is then used in the vaccination. Key feature is assurance that there is no toxin activity in the end product that would be a safety issue for the vaccine recipients. This is true whether it's a human or veterinary vaccine. The WHO guidelines include testing at different stages of development and production of a vaccine to assess the presence or absence of a toxin. And these tests traditionally have included an animal-based method. We know now, however, that there are opportunities to apply 3Rs to these animal tests. So the main strategies to address 3Rs for toxicity testing include replacement with an in vitro approach, reduction by eliminating redundant tests, and to evaluate the relevance of tests and to remove a test if there's no added value. So product groups and specific products can be evaluated using these principles, and all of the strategies depend on the use of a well-characterized and a well-standardized production process, together with the added value of the in-process quality control tests and monitoring of the products and the related intermediates at the appropriate stages with the relevant tests. So you really have to look at the whole picture of the process and the testing scheme to see what the best approach is. In this slide, you see the WHO requirements for toxicity testing of diphtheria, tetanus, and acellular pertussis. In the first column, the tests to be performed in the bulk purified toxide, then the final bulk, and then the final lot. And you'll see that there's testing in both the bulk purified toxide and the final bulk for all the three DT and AP. Specific toxicity testing and reversion of toxicity in the bulk for diphtheria and tetanus with guinea pig tests for absence of toxin, and reversion of toxicity again in the guinea pigs for specific toxicity and for reversion of toxicity. In the final bulk, specific toxicity again in the guinea pig model in both diphtheria and tetanus, although it does say that it may be admitted once consistency is demonstrated. And then for acellular pertussis, the HIST assay in guinea pigs or the CHO assay for the bulk purified toxoid. And in the final bulk, again, the residual activity of pertussis toxin with the guinea pig assay or another suitable assay, and the reversion to toxicity again with HIST, so the guinea pig assay. In the last column, you see each of them in the TRS includes the innocuity test, but it's very important to remember that in the ECPS 69th report decided to remove the innocuity test and the abnormal toxicity testing for biological products, and this is recorded in TRS 1016, 2019. This is an excellent example of removing a test with no added value. So these are the requirements in the WHO TRSs. Let's look to the different possibilities that I expressed in the earlier slide. So in vitro possibilities, what are there? For diphtheria, a special and highly sensitized non-animal method, the virocell assay, is available for the detection of diphtheria toxin. You can see the reference to the method there, and the method is also described in the WHO Manual for Quality Control of Diphtheria, Tetanus, and Pertussis Vaccines. We know that use of this method is not possible on the final bulk stage due to the presence of adjuvant, but this can be dealt with when we evaluate the whole process, and you'll see that in the next slides. So tetanus, there's an in vitro opportunity under evaluation, and in fact, the study has been completed. We're going to speak briefly about the binnacle test in this presentation, and I know there's another presentation on it later in the program. So you'll get to hear more about that. For acellular pertussis, there is also a specific and sensitized cell-based method, the CHO cell assay, that takes into account the receptor binding, translocation, and enzymatic activity of the pertussis toxin, and it can be used to detect pertussis toxin. So this is a good alternative. There are standard protocols that have been developed. There's some references there you can see, and there's both an direct and an indirect version of the assay for testing on the final bulk vaccine and also in the presence of adjuvant. It's important to remember that any alternative test needs to be specific. It needs to take into account the key actions of the toxin and be at least as sensitive as the existing animal model so that there's still the assurance regarding the safety of these vaccines and that is maintained, which is the key goal. So the other possibility is the elimination of redundant tests. And what I mean by redundant tests is that sometimes tests, as we saw, are done at multiple levels. So according to the WHO TRS for the DTwP vaccines, the tests for toxicity are potentially performed at 3 stages. It may be performed during validation and during development. It might be performed on the purified toxoid, or it might be performed on the final bulk. Remembering, of course, that for D&T, this test could be removed once consistency was established. And for pertussis, the test for aversion can be omitted if validated during development. So it's important to assess the need for the test at each step, considering the ability of the remaining tests to assure the overall safety to the same degree. Is the test done at one level, either development or validation, or the level of the purified toxide sufficient to cover the requirement of safety for the whole process? And finally, the relevance of the added value of a test. So we've already seen the example of the innocuity or the ATT test, where there was significant evidence accumulated to demonstrate that the test does not provide added value. And there's some references down there if you want to look into that in more detail. It's a historic test not designed for the actual purpose. And as I mentioned, the removal was endorsed by ECBS in October 2018. Another example is the test for reversion to toxicity of the tetanus toxoid. Looking at the scientific relevance of the test design, testing for reversion to toxicity includes a test in guinea pigs on test samples stored at 34 to 37 degrees and control samples stored at 2 to 8 degrees for 6 days. However, evidence from the development of the binnacle method in the BSP program and by the project leader demonstrated that the tetanus toxin rapidly loses activity at 37 degrees. So storage at 37 degrees for 6 days really didn't make a lot of sense. So this allowed the opportunity to reevaluate the need for this test. There was also historic evidence that there was lack of reversion, and this was deemed from the review of data from production records. So we look at the pharmacopoeia and how we've applied all of these different strategies. You'll see for the diphtheria, tetanus, acellular pertussis, in each case, there's a test done at one level. So for diphtheria, it's on the bulk purified toxide. Same for the, well, in fact, they're all done on the bulk purified toxide. The tests during development have been removed, and the test on the final lot for the acellular pertussis has been removed, because the remaining tests done on the bulk purified toxide were considered to be sufficient. In addition, we have the removal of the irreversibility of toxoid for the tetanus vaccines, which I mentioned, and also the acellular pertussis. And of course, the abnormal toxicity test is completely removed from the pharmacopoeia. So here we have the examples where there's no more in vivo testing for diphtheria, for acellular pertussis, and for the tetanus, we still have the guinea pig assay, but there is an important opportunity that is now available and can be investigated. And that promising new possibility for the tetanus toxicity is an in vitro test. Was run in the biological standardization program and under the project code BSP-136, and the project leaders were Heike Berendorf-Snickel and Beata Kramer from the Paul Ehrlich Institute, which was the OMCL who developed this assay. Binnacle stands for binding and cleavage. It's a two-step assay, fully in vitro, and it's based on the toxin function with the binding and the cleavage, and it completely replaces the guinea pig assay for a certain toxoid. So the study is completed. The transferability was shown to be successful. There was good repeatability and reproducibility, and the limit of detection was consistent with the guinea pig test. Product-specific validation will be required because it's not suitable for all toxoids, but it's suitable for many and is a good alternative for those. The European Pharmacopoeia Expert Group for Human and Veterinary Vaccines is now preparing for inclusion of a reference to the Binnacle method in the monographs. And we have 2 publications that are available free online in the EDQM online journal. You can see here. And there was an EDQM webinar on the 12th of November in 2024. And the resources from that, including a full recording of the presentations, and the presentations are available on our website if you want to look at more into that. Just a few words on wholesale pertussis. So this is covered by WHO TRS 937, Annex 6. Bacterial inactivation and detoxification process has to be validated to ensure the absence of toxicity. There are controls of the single harvest. You can see here, no biologically active heat label toxin should be present or detectable. The method of detoxification should ensure safety. Limits aren't recommended for each toxin, endotoxin, cytotoxin, and this has to be appropriately validated by the manufacturers for consistency. Control in the final bulk. There's specific toxicity testing with the mouse weight gain test, but there is also a possibility to monitor pertussis toxin using the CHO cell-based assay. And of course, as with the others, the innocuity test has been removed from the final lot. So in conclusion, there are opportunities to reduce the use of animals for toxicity testing of toxoid vaccines. EDQM is committed to the application of state-of-the-art approaches for the quality control of medicines, always keeping 3R considerations embedded in our strategies. And we have made significant progress over the years to reduce, replace, refine, and remove tests involving animals, including DTwP vaccines. All of this work is achieved through the good work of our community and our experts in the European Pharmacopoeia and the Biological Standardization Program, supported by OMCLs, other experts and stakeholders, including industry partners. It's important to remember that all decisions for change are based on sound scientific principles and consultation within the wider community. And we will, of course, continue to assess opportunities for advances in 3Rs and improve analytical tools. Continued effort is needed to have a harmonized approach, and global acceptance of effective and robust alternatives is a key to making sure that these scientifically robust and relevant methods get implemented at a global level and are applicable. And with that, I thank you.
Thank you very much, Catherine, for this important overview that is really setting the scene about all the available opportunities. And it's good to see that some of them, they have been recently developed, while others they are around since a little bit of time. So it would be interesting to hear from the attendees what is their experience, if they were successful with some of them. So thank you, Catherine. Please just add your comments and questions within the Slido. Right now, I would like actually to open up the other piece of this workshop that is actually going into providing you some more information about the opportunities for diphtheria, tetanus, and the wholesale pertussis safety testing. The first speaker of this session is Dr. Pradeep Das from Biologically, and Dr. Pradeep is actually going to give an overview about the experience of Biologically in using the VeroCell assays for the test of the absence of toxin and reversibility of toxoid. So thank you very much, Pradeep, for being with us. Your slides are coming up immediately. Please let me know if you have any issue in seeing the slide moving. Thank you.
No, I can see the slide. Thank you, everyone. And thank you, Laura, for giving me the opportunity to share my experience in going from in vivo to in vitro, particularly on diphtheria, absence of toxin, specific toxicity, and irreversibility of toxoid in viral cell assay. Can you make it in the slide mode?
It is. You cannot see in the slide mode. So give me a second. So
No,
I'm
and
trying
in fact,
to
Laura,
recreate.
we didn't see your slides in slide mode either, so we didn't see the QR code when you were sharing it initially. So you might want to
Okay.
do that again at some point.
I will try.
Okay.
I will try.
Okay. So give me a second. That's I'm going to ask Heidi to do that. Okay.
Can you see the slides?
Yeah, it is coming in duplicate.
Now we see it in the presenter's mode. I think you have to switch screen somehow. We've got the slide plus the panel on the side.
Is it not? What do you see now?
Yes, I can see, but it is not in the slide mode. If
Nothing.
everyone can see that, maybe we can continue. I'm okay with this.
It should
In
be.
presentation mode, if it comes.
Now, if you go up to the second, you see there's three little, I don't know what it says, it's in German. On the top in the black, there's one, the second one, it will allow you to switch which screen you're showing.
In the second, not this one. If you make it in the slide mode in the last part of it, where the percentage of font is there. No.
Sorry.
Apologies for these technical issues. Thank you also for the recommendation from the chat.
What do you see currently?
Nothing,
Screen?
Irina, we cannot.
Shall I share my screen? Let me try.
If you can, Pradeep, please.
Yeah.
Sorry for that.
Yeah, no problem.
Let's see. Okay, we can see your slides and
Can you see my
presentation mode.
screen?
Thank you, Pradeep. You can go ahead. Thank you.
Yeah. Thank you, everyone. And once again, thank you, Laura and the team, scientific community, for inviting me for this one of the special issues on the diphtheria, absence of toxins, specific toxicity, and irreversibility of toxoid on viral cell assay. So as per the WHO TRS guidance, we have the detoxification stage, specific toxicity, and reversion to toxicity stage, where you need to perform the test like detoxification test and the specific toxicity test, where the toxicity is for the presence of diphtheria toxin is to be performed. And currently the test is performed in vivo using guinea pig or in vitro by any suitable cell culture assay. And there is one more topic is called reversion to toxicity. So in this case, each bulk purified toxide should be tested to ensure that reversion of toxicity does not take place during the storage. The test may be performed in vivo using guinea pigs or in vitro by using suitable culture method like virocell assay. And the detailed procedure is already mentioned in the WHO manual for quality control for diphtheria, tetanus, pertussis vaccine, which is WHO IVB 11.11, European Pharmacopoeia 11.2, and Indian Pharmacopoeia 2022. So how the test is carried out, if you can see the comparison, it is an in vivo test wherein for the absence of toxin, which is called specific toxicity, is performed in 5 guinea pigs, 250 to 350 gram, and you inject 500 LF of the drug substance by subcutaneous route. And for the irreversibility test, you take the animals of greater than 500 gram, and you inject intradermal route with 50 LF concentration and 0.2 LF concentration of each at different temperature conditions like 228 and 34 to 37. However, all these tests are required with different number of animals. In my future presentation, it will come. Now, all these assays comes in the right side of the presentation, which will come in 196 well plate, wherein the first part, which is related to, we are taking this as a fresh sample, which is for the specific toxicity directly, which is diluted to 100 LF. And you make 3 serial dilutions of 100 LF, 50 LF, and these all are in triplicate. And similarly, for the reversion to toxicity, you take 2 to 8 degree and 34 to 37 degree in the similar instances. And the last 11th and 12th row, you make this as a cell control. One is blank with media, one is DAT, which is diphtheria antitoxin control. And this is your control toxide, which is already validated, which meets the testing requirement. This acts as a positive control. So this part of the assay is run for the specific toxicity, and the second is just replica, wherein we add the diphtheria antitoxin. So when you add this diphtheria antitoxin, suppose this toxin is present in the first series of this, here all the cells will die, and because of available of diphtheria antitoxin, it will neutralize here, which will confirm that yes, it is passed with the it has the diphtheria toxin in the drug substance. So the criteria is very simple. With a 96 well plate, you can eliminate complete testings, and this acceptance criteria. The defined is the geometric mean of optical density OD of the cells containing viro cells together with the control toxide should be calculated and divided by 2 to obtain 50% OD value. So it is an automated process. Once you do the exercise, you just take the OD values, and based on the control toxide, your reference, your sample value OD should be greater than that. So that meets the your testing criteria. And as per the Indian pharmacopoeia and European pharmacopoeia, and the test is invalid if 5 into 10 power 5 LF concentration of reference diphtheria toxin in 100 LF per ml toxide has no cytotoxic effect, where the test is invalid. As per the WHO IVB 11.11, the test assay is valid if sensitivity and specificity of the assay is confirmed, and the control toxin is toxic to viro cells which is neutralized in the duplicate wells containing toxin and antitoxin. So this part, which contains DAT and your toxin, it should neutralize. The point number D and F is your toxin part, which contains only toxin and media, and this is as a reference where you can identify how much is the concentration of LF, which the toxin can neutralize the virocell assay. So this is a design of plate layout. Once batch is passed and if the batch is failed. As you can see over here, once the batch is passing, here all the cells are having greater than the positive control, and it is meeting the test requirement, which is very sensitive and specific. However, when the toxin is there, when the batch is failing, you can see there will be completely cytotoxicity. And it is because of this DAT, E, F, and G column, it is neutralizing the toxin because you are using DAT, diphtheria antitoxin. So here it shows that it is confirmed that your drug substance has a diphtheria toxin in it, and it is neutralizing by diphtheria antitoxin. So this is a pictogram of where you take the OD values, and if you can see, this is the past results where 0.61 is your control toxide OD values, which is divided by 2 as per the arithmetic calculation, 0.30. And you can see your test sample should be greater than this 0.30. So if you see here 7.43, this all area it is greater than 0.73, but it is more than the required number. So it is meeting the test required. But when the test substance is failed, you can see the OD values, it is gone beyond the less 0.3. So this product becomes fail for the drug substance. So it is very, very sensitive. And side by side, I put the pictures also, how the color changes. Visually also you can identify you can calculate, but it is always good. And as part of 21 CFR compliance with the plate reader, this removes the manual intervention. So it will be very much robust, and you can rely on your data with more accuracy and precision. So this is one of the overall summary which I want to emphasize on how the diphtheria currently. If you see this, I have mentioned like specific toxicity, you go with the guinea pig. Only the sensitivity of this, your substance is 0.1 LF. So if it is less than 0.1 LF, the toxin is there, animals cannot able to detect. And there is always chances of repeatability because of invalidity. Hence, there is always much more complexities are involved when you use the guinea pigs. But when you virocell, if you perform in one single plate, where the sensitivity you can see it is 3 to 10 power minus 5. Similarly, the case of irreversibility, where you perform the intradermal method and of course subsequent method and compare to virocell, you can find the intradermal is this type of reactions, which is very, very painful to the animal. And its sensitivity is 3 into 10 power minus 4. However, in the viro cells, when you perform 3 into 10 power 5 LF is the sensitivity. So in one plate, all the three tests like specific toxicity, irreversibility of 228 and 37 degree can be combined together in just one plate. So you need not to have, which is this assay has more sensitive compared to all your assays related to specificity versus irreversibility. So in conclusions, we know that the animal's requirement is always a challenging, and it requires more number of days. And welcoming through the in vitro method, virocell assay, it is more sensitive and reliable. And results observations is by ELISA reader. There is more manual intervention, and no animals are required for utilize. It is a complete replacement of the assay from in vitro to in vitro and very easy to use. That's all my presentation for this. Thank you.
Thank you, Pradeep. Since you might not be available later, we have a question in the chat that is about an experience for the Clostridial toxoid potency by total combining power. Is it something that you had a look, or are you familiar with this test?
No, I have heard of this, but I have not gone through that complete total combining power list.
Okay, thank you. Thank you. Okay, then if someone else has any experience, we can go back to this during the discussion. Thank you, Pradeep, for your presentation.
Thank you.
Okay, and now we come to the next presentation. It's about the binacle assay, this very famous binacle assay. The presentation will be held by Shahjahan Shahid from GSK. Shahjahan is an expert with respect to triage and know him for a long time now from the Vec2Vec project. Shahjahan, I'm happy that you present. Please, the floor is yours.
Thanks a lot. I will try to share my screen. Give me a second. It seems to be not as easy as I thought. Can you hear and see me well in the meantime? Oh, can you hear me?
Yes, we can hear you.
Okay, that's great. Then I will try to share my screen. You should hopefully now be able to see my screen.
Yes, it is here.
Okay, perfect. Switch into presentation mode. Hi, so thanks for the nice introduction. And indeed, we worked together for a long time. I work for GSK Vaccines, and I'm here today to present you the Binacle project that was already introduced before by Katrin. Thanks a lot for explaining the essay. What you will see here today is the same presentation I gave at the EDQM workshop linked to the applicability of the essay. And what we're busy with in GSK to overcome the challenges to bring this really into everyday use. Before I go a bit more into details and what we are currently busy with, I would like to highlight that in GSK we are committed to animal replacement, especially in quality, where a lot of our animals are used. We have set as a target that we will reduce the animals by 75% in 2025 compared to 2015. So far we're at 66%, and so by end of the year we aim to reach the 75. If we would go back into the past another 10 years, the animal reduction achieved would be even much higher than this. But now let's jump on the topic and the discussion of today. And this is linked to the tetanus vaccine and the required safety test. As you are probably aware, tetanus is a very potent toxin. It's even listed as a biological warfare agent because it's that dangerous for humans. And therefore, it's of course very important that a proper inactivation and the required analytical method is in place. What you see in the left in gray is a simplified manufacturing stream of our tetanus manufacturing. So you start at the working seed, main culture. The bacteria then produce the toxin, which is harvested, inactivated, filtrated, blended with other components, and you get your final product. As you also can see, what is shown in blue are the different animal tests that were in place to ensure the safety. And there were really only animal tests in place to ensure the safety of tetanus. All of these tests have been removed, and this is really thanks to the great collaboration with the EDQM that are pushing out those animal tests the moment data becomes available that they are no longer required. What we are left with today is what is shown in orange. That's the absence of the tetanus toxin test. This test has been selected kept because it's the most sensitive compared to all the others, and it therefore ensures the best safety for the patient. How the test works is basically you inject 1 milliliter of your toxoid into 5 healthy guinea pigs. They are observed for 3 weeks. If they show no signs of an intoxication, meaning symptoms of tetanus, the test is considered as pass. If we now want to replace this test with the Binacle, the most important thing is, of course, the sensitivity of the assay. With all the other tests gone, it means the in vitro assay, in this case the Binacle, has to be at least as sensitive as the absence of tetanus toxin. And this is the challenge, of course, for the Binacle assay and also from us, from an industry side, to reach. How the Binacle works is 2 ELISAs, which are conducted consecutively. You have the tetanus B chain, which binds to a ganglioside. That's your first ELISA. Then whatever is bound is moved to a second ELISA plate, where you then assess if the tetanus A chain, which is kind of an enzyme, cleaves the synaptoprevin. And this is then also the damage tetanus does to the human body and resulting in the muscle tension. When we conducted the BSB study, we had really very positive feedback from the 3 technicians in our laboratory. They were very pleased with the SOP on the handling. Things went very well, and that was for us basically the trigger to move ahead and say, can we not make this a standard QC test? When we started with this activity last year, and we encountered several challenges that I would like to share with you, and also how we try to overcome them. And I really have to say to try, because today we have not reached yet the final resolution for them. The first one, and that's for me the most critical, is linked to a stabilizer called TMAO. This is an osmolyte. You add it into your ELISA plate. It helps to increase the interaction between the tetanus toxin and your proteins, meaning it increases the sensitivity of the assay. And as you remember, the sensitivity is really going to be one of the key criteria to decide if the Binacle can replace the guinea pig test. What we have seen, and this is the challenge, and what is also shown here in the graph, is that when we purchased 2 batches from 2 suppliers for TMAO. And so TMA 1 and 3 is from one supplier, TMA 2 and 4 is from the second. And we have seen quite some difference in the performance from the same supplier. So there seems to be a quality issue in those TMAO. We try to understand if it's contamination, did some investigation, talk to the suppliers. This was not really successful. So we are now looking into alternatives. We did quite an extensive screening, which you can see here. So we always checked from 2 suppliers alternatives. The 2 most promising are sarcosine and beta-ine. They have quite a similar chemical structure as TMAO, which therefore we also thought they could be the most promising. In the current setup, because we still use the setup used in the BSP, they are not as sensitive as the TMAO. Our next step will now be to understand if we modify the original BSP setup, if we can improve the sensitivity to ensure it's better than the animal test. This is, for me, really the most critical challenge we are facing. The second one is then linked to the antibody. So because the Binacle assay works as an ELISA, at the end you need an antibody to quantify your analyte, which in this case is the cleaved synaptoprevin. You can see the synaptoprevin here at the bottom left, and you see where the orange arrow is where the tetanus A chain cuts off the synaptoprevin, and then you need an antibody that only binds to this cleaved part of the protein. In the BSP study, a rabbit sera was used, and it worked very well. So it was very specific and sensitive, which is in fact quite unusual for rabbit sera or polyclonal sera in general. From a GSK perspective, that is not ideal for several reasons. First of all, it still relies on animal use. So would continuously have to inject it into rabbits and bleed them to get the reagent. Secondly, it does not ensure that if I generate a sera today, in 10 years, it will be still as sensitive, because that, of course, depends on the individual animal. And the third issue is when we try to mimic what was done in the BSP study and use the same supplier, that was quite a challenge to finally reach this reagent. Therefore, from the very start, our idea was that we will instead use a monoclonal antibody. Our first idea was to purchase those. However, the one we got access to were not as sensitive as the one in the BSP study. We then generated animal-free monoclonal antibodies using the phage display system. However, they were not specific enough. So finally, then we moved to a mice hybridoma monoclonal antibody. This is what you can see in the graph on the right. So in the red, it's the rabbit sera, as in the BSP study, and in purple, the monoclonal antibody. Again, we still use the BSP setup, so the sensitivity of the monoclonal antibody is not as good as the sera. And the next step is similar. We need to optimize the assay now with the TMAO replacement and the monoclonal antibody to see if we can get the proper sensitivity. So these are the 2, I would say, technical challenges. Then we also have 2 compliance challenges, because we work in the current good manufacturing practice system. This is a requirement that is not negotiable, because we need to ensure the right quality for the patient. Therefore, each reagent we have to use have to have the right standard in terms of quality. The recombinant synaptoprevin, so if I just jump back a slide, so remember the protein that in the end is used to make a readout and to understand is the Binacle sensitive enough, is today not available by a GMP manufacturer. So today it would not reach the right quality. The vendor used in the BSP study did a great job, but however, it is a small vendor that does not have the standard that is later then required to make this a release assay in quality. This will not be accepted by GSK, neither by our controlling bodies. And therefore, we need to identify another supplier that can fulfill the standard. And then, of course, GSK is not the only tetanus manufacturer in the world. So I think, therefore, it would really make a lot of sense if we can identify 1 or 2 suppliers that can ensure the supply more globally, and that there's an alignment between the different manufacturers about small but crucial details. Because in the BSP study, the E. coli expression system was used. So should we then stick with this one? Because if the next one goes to a yeast expression system, you will have different glycosylations on your protein, which could modify your sensitivity quite a lot. That's one element which I think can be overcome with the right organization. The second one is about the assay requires a positive control. So because our toxoid is not toxic, so what we want to see is no reaction, but you need to have a positive control to show, yes, it does. My assay does work. It can detect tetanus toxin with the right sensitivity. There's no positive control in place in the today's safety assay, because that is, first of all, not required and would be a waste of animal lives. And therefore, one needs to be identified. We purchased 4 different tetanus toxins. There were quite limited information available, so we do not know well how pure they are, how old they are. What we have, however, seen is there was quite a difference between the 4 we purchased regarding how well they were detected in the assay. And that seems also to be linked to how the purification is done by the manufacturer and how they determine the amount of tetanus neurotoxin. And I think here, therefore, it's also quite important to align on how could such a standard look like, because all the manufacturers that want to switch to this assay need to identify one and need to ensure that once the batch they have selected is empty, they have a procedure in place to bridge. And if this is not aligned, we will end up with very different sensitivities by different manufacturers, simply due to the standard that was selected. And that will then cause questions on why is one assay performing differently than the others? Are you sure it's really more sensitive, etc., etc. So I think this is also quite important to have an alignment. The third element, and this is not a technical one, is the driver here is clearly the European Pharmacopoeia, which is the most progressive in animal replacement. And I would really say thanks to the European Pharmacopoeia, we do see a drop in animal testing in vaccine manufacturing. Our products, however, are not produced only for the European market, so they are sold worldwide to all the patients. And it's therefore quite important to understand what are the requirements from authorities outside of Europe to understand what they want to see to accept such an in vitro assay, something like the Bionica. What we can imagine, but what really needs to be clarified, is what is the role of the matrix impact, so the interference coming out of each particular manufacturing process, because you might use different strain, different bacterium media. The limit of detection, especially when it comes to the reference toxin that is used, so your positive control, again, in the respective manufacturing matrix, and what would be really required to convince them in a comparability study between in vivo and in vitro. So far, the gold standard remains the in vivo assay. However, the limit of detection has never been described for this assay. When you look into the literature, you will only find very old publications from the 1950s, from the 1960s. And that means at the end of the day that for the manufacturers, to determine the LOD in vivo is very likely a requirement to convince authorities around the world. That brings me to my last slide, where I just want to conclude that with everything said today, so today the Bionica is not ready to replace the guinea pig assay. However, I believe that tomorrow it can be ready, because the Bionica shows really great potential. The technical elements, I think, can be overcome, as well as other elements which simply requires alignment and agreement by different manufacturers and by the controlling bodies. And with this, thanks for your attention. And I think the questions will be at the end of the talks, right?
Thank you. Thank you. Thank you, Sajan, for your presentation and sharing a very concrete approach to the binacle and the complexities that are still in place and needs to be overcome. It will be a very interesting conversation afterwards, really, to show exactly what does it mean to invest on a new methodology and bringing into the QC routine. So thank you very much. Please just drop any questions to Sajana in Slido or in the chat. And I would like to move to the next presentation that is also going to present another methodology that is currently in development. And it's again for tetanus, and it's a cell-based assay. And to present this opportunity is going to be Dr. Andrew Peden from the University of Sheffield, presenting the work that he's doing together with his colleague Chara Duran at the Sheffield University, and also with Shalini Rajagopal from MHRA. So Andrew, the floor is yours. Thank you for being with us.
Thank you. Can you hear me and see my presentation?
Yes, Andrew, you can go ahead. Thank you.
Okay, so I'd just like to thank the inviters for coming, inviting us to come and give a talk today. And so we're going to be talking about the work we've been doing to develop novel cell-based assays for testing the biological activity of clostridial neurotoxins. And today it's obviously going to focus on tetanus toxin. This has been a very long-standing collaboration between the MHRA, the Medicines Healthcare Products Regulatory Agency, and the University of Sheffield. And so we really would like to thank them for helping advise us to try and sort of translate our work from being sort of very basic and fundamental to something that's a bit more applied. So thank you for the previous speakers. I don't think I need to dwell too long on this. Obviously, there's a lot of animal testing historically being used to sort of measure the biological activity of clostridial neurotoxins. Obviously, they're the most potent toxins found in nature. So obviously, there's a large amount of safety testing required to ensure that either measuring their potency if you're making botulinum toxins, or actually ensuring that there's no residual toxicity if you're making vaccines, which was the sort of key focus of today. And obviously, as we've mentioned, it's been mentioned, obviously, one of the big challenges facing, manufacturers that animal work is often there's large variability, which leads to requiring to use large number of animals. And obviously, these assays are very time consuming, so they can take quite a long time, anywhere up to 21 days and even longer for some of these assays to ensure there's no toxicity or residual activity. So my group at the University of Sheffield and the collaborator who started this project, Bazbek Dasletov, we were interested in the basic biology of botulinum and tetanus toxins. And so most of you will be aware that botulinum toxins enter neurons or motor neurons at the neuromuscular junction, and basically they're internalized. They then escape into the cytoplasm of the neurons and they cleave the snare molecules, so the VAMP or the synaptobrevin, which drives synaptic vesicle fusion. And this causes flaccid paralysis, where tetanus toxin, on the other hand, is still taken up by the motor neurons, but retrotranslocates to inhibitory neurons, where it then has its mode of action. And this is why you get a rigid type of paralysis if you get infected with tetanus. And so we're labs, we're spend a lot of time understanding this basic biology, and we wondered whether we can use this our core skills to try and develop novel approaches for actually measuring the biological activity of these molecules. And so just to sort of remind people that when these toxins, so for botulinum B and tetanus, when they are released into cytoplasm, the light chain or the protease domain of the toxin targets this molecule called VAMP or synaptobrevin, VAMP2, and basically it chops the protein, so it's molecular scissors, and this inhibits the fusion of the synaptic vesicles at the neuromuscular junction. So we were approached a long time ago, over 10 years ago, by Thea Sideric and Paul Stickings, who work for the MHRA in the UK. And we were basically challenged, would we be able to use our sort of knowledge of the biology of these toxins and the snare biology to try and come up with some form of cell-based assay which would be useful for manufacturers. And so with these assay, this type of assay, would it be useful for testing the potency of, say, botulinum toxins? Could it be used for residual toxicity testing with vaccines? And then actually, could it be used for testing antitoxins? And I'm going to show you where we sort of reached in the development of these cell-based assays. So there's three components to the cell-based assay that we've developed. And I should say that one of the reasons we've gone for a cell-based assay is it completely recapitulates all the biological steps of intoxication. So the binnacle assay only measures binding and cleavage, where our assay measures binding, endocytosis, endosomal escape, and translocation. So completely representing all the biological steps or the physiological steps of intoxication. So in our assay, we basically have neuroblastoma cells, and we've identified several different types of neuroblastoma cells which are sensitive to botulinum and tetanus toxins. We've engineered them to express a recombinant reporter molecule which has a nanoluciferase tag. So it means we can simply measure the activity of the toxin using a luminescence-based reaction. And then finally, which you'll sort of be familiar with the binnacle assay, we have a cleavage-specific antibody. And at the beginning, we used a cleavage-specific antibody, which was given to us from the MHRA. But I'll show you some slides that we've generated our own one. And so in this assay, we take neuroblastoma cells, we differentiate them for 3 days, we incubate them in the toxin for a further 3 days, we lyse the cells, and we do this one-step ELISA reaction, and that will give us the output of the cleavage of the VAMP molecule or the activity of the toxin. And so when we first set this up, and this is work which was done in collaboration, it was done with Kira and my lab and Shalini at the MHRA, the assay we developed wasn't actually any good for measuring tetanus toxin. The cells that we developed weren't sensitive to toxin, but actually they show good specificity to botulinum toxin. And so you can see here we have the control, the non-treated sample. We treat them with the botulinum B, and then if we use the WHO antitoxin, you can see that we can completely abolish any signal showing the specificity of the assay. So through additional work through Kira, we were able to actually develop a neuroblastoma or identify a neuroblastoma cell line or population of cells which were sensitive to tetanus. And part of this work was published as VAC2VAC. So we've transferred the tetanus toxin sensitive cells to the MHRA. And then what they were doing in this study, they were testing different anti-tetanus toxin antibodies and also the toxoid. And so you can see, we see specific signal in our assay when we incubate the cells with tetanus toxin. And if we add the different antibodies, we can nicely abolish this interaction. And so this was quite promising. This suggested that we had a cell-based assay which potentially could be useful in this sort of manufacturing space, but we realized there were some significant limitations that we needed to overcome. And as pointed out by the speaker from GSK, obviously this assay used a rabbit polyclonal antibody, and we realized that this antibody, this sort of security of using a rabbit polyclonal just wasn't going to be sort of secure enough for going forward with this assay. So basically what we've done is we've made recombinant capture antibodies which see the cleaved VAMP molecule. And so I can show you this in this slide here. There's a little bit of validation. So this is basically our cells which have been treated with botulinum toxin. I should say the cleavage site for botulinum and tetanus toxin is the same. And you can see that we only see signal in the samples treated with the botulinum toxin. So these antibodies are highly specific, and we've done a lot more characterization than I'm going to show today. So we've done a lot of work on trying to obviously improve the capture antibody, so having a sustainable capture antibody. Through work with the MHRA, we're developing SOPs to improve the assay. We've also identified alternative neuroblastoma cell lines which are sensitive. And so what I'm just going to show you, this is a snapshot of where we are with the assay at the moment. And so in this experiment here, we've basically treated the cells with basically increasing concentrations of tetanus toxin. So this is the non-treated control. This is 100 femtomolar, 1 picomolar, and 10 picomolar. And so what you can see is that we have very good or sort of comparable sensitivity to the guinea pig bioassay. And it is true, the literature regarding the limit of detection in guinea pigs for tetanus toxin is quite variable. So there's some studies saying it's 1 to 2 nanograms. However, we've gone with a study which is saying it's down at 0.1 nanograms. So we can see basically our assay shows comparable levels of sensitivity. So this is all very promising, but the question is, can we translate a sort of research grade assay to an assay which is useful for manufacturers? And so we realized that, you know, this was going to take quite a bit of work. And so we were very lucky. We applied to the NC3Rs in the UK for funding, and they funded us to perform a global, basically a sort of assay to sort of investigate the sort of transferability of our cell-based assay. And so at the moment we're in phase 1. And so at the moment we've got 10 tetanus toxin manufacturers, toxoid manufacturers, who have donated samples. So 8 of these are human and 2 of them are veterinary. So those samples are being donated to the MHRA. They're then being anonymized and then basically transferred to the University of Sheffield, to my lab, where we're testing them to see if they're compatible with testing in the cell-based assay. There's experiments also being performed at the MHRA to sort of confirm the studies that we're also performing. And then if things go well, we'll move into phase 2, which we're hoping would be later the end of this year, where actually the cell-based assay would be transferred to manufacturers, where they can do in-house testing and validation of the assay and testing the critical reagents. In addition to this, as it was sort of pointed out by the speaker from GSK, we realized we need a tetanus toxin reference reagent, because obviously, you know, one of the challenges is how everybody will validate their cell-based assay or even the binnacle assay. And so we've been very kindly donated some sample from one of the manufacturers, and the MHRA are working to investigate whether they can turn this into a reference material which they'll be able to make available. And I was going to show you a little bit of data from this in a second. So we're still in stage 1, and it's taken quite a long time to sort out all the legal paperwork to get all the 3-way MTAs in place. And so I can say we now have 10 manufacturers, and we have another 2 manufacturers are coming on board to the study. And so we're going to have 3 tranches of toxoid testing, and I'm just going to show you some very early preliminary data. And I should say this is, you know, we still need to do quite a lot of work on this. And so as you've seen in the other talks, the standard assay, a guinea pig would be injected with 500 LF. And so we've done various different titrations of some of the toxoids we've been donated. And we've also compared to the WHO reference toxoid sample. And so you can see the WHO standard or control, we can go up to 200 LF and the cells are completely happy under these conditions. However, you can see that for other toxoids, basically, we see significant cell death, even at lower LF levels. And so one of the things we're working out at the moment is how we can overcome this. And so for some of the samples, basically, you know, if we dialyze them, we definitely see significantly improved cell viability with the toxoids. And so this is something, so maybe we're going to have to do some work on each different toxoid to get it to a format where it'll actually work in the cell-based assay. So the question is, you know, what do we see when we put these toxoids on our assay and measure cleavage of VAMP or response to the tetanus toxin? So this is an example. So this is using the WHO standard. So this is our non-treated control. Here's 20 LF and here's 200 LF per mil. We see no signal at all, suggesting that it's been obviously should be completely inactivated. As our tetanus control in this experiment, we've used some of the manufacturer donated sample. And so this has got potency units. So we know that the potency of this toxin, or the limit of detection, is 10 to the -4, between 10 to the -4 and 10 to the -5. And in this experiment, we've used 10 to the -4 of the toxin. And you can see that we see a very robust signal from this toxin. So hopefully this is just giving you a little bit of snippet of a sort of an alternative cell-based assay or alternative assay to the binnacle which we're developing, which is potentially showing signs that will be useful for some, certainly for some toxoid samples. So just to summarize what I've shown you, so the cell-based assay is relatively simple, it's quantitative, works in an ELISA-based format. Most importantly, for some regulatory environments, it covers all the biological steps of intoxication. It's obviously far faster than a standard guinea pig test. It's scalable and IP protected. So we've patented the cell-based assay, and we're now we've filed patents to protect the recombinant antibodies that we've generated. And actually, our data suggests that some of the cell-based assay will be suitable for some certain toxoid samples. One of the things that I forgot to say is that to try and help the translation of this cell-based assay, we formed a company called Sansanima, and so the postdoc Kira Doran, who I showed you some of her data, is now the CEO, and I'm a chief technical officer of this. And so we will try and use this to try and help overcome some of the problems which are being faced to try and standardize the development of this assay. So finally, I'd just like to thank the people involved. So obviously, Kira has been a long-standing postdoc who's worked on this project for over 10 years, who did initially some of the pilot, the original work, and then obviously Jada has taken over the baton, and Charlotte Lees is doing some of the biochemical experiments. And then obviously this work really wouldn't be possible without the input of Paul Stickings and Shalini, and they've really helped guide us and actually set us the challenge to try and solve, and really sort of trying to help us translate the basic cell biology into something which potentially could be useful in a commercial environment. And obviously I'd like to thank Thea and Basbek, who initially started these projects. So thank you very much for your presentation. Listen to my presentation.
Thank you very much, Andrew. There is the request to go get the publication that you mentioned. So if you can just drop in the chat the link to the correct one. And then there is also a quick, I hope that is a quick question about your knowledge on whether your test also works with other clostridials like the perfringent type D, the septin cone, the Novi type B.
Yeah, so at the moment, no. So our assay is basically it's designed for sort of botulinum potency testing, so related to botulinum B or for tetanus. But I guess in the future we could maybe try and develop these assays, but it's not what we're doing at the moment.
And then just the second part of the question is that the LD50 is the LD50 the total combined power test?
I'm not sure actually. Maybe that Shalini might be able to answer that.
Okay.
I'm not sure on that one either. I'd have to take that one offline. Yeah.
Okay, so thank you both. If you can double check, and thank you, Natalie, for your question. So, okay, again, thank you very much, Andrew and the whole team for sharing the results of your work, which we are appreciating to see exactly how does it mean to actually find additional opportunities in terms of method development. We would like to go back to, please, Idemarie, please.
Sorry. I just wanted to introduce the next speaker.
Please, Idemarie, go ahead. Thank you.
And with this, I now go again to Dr. Pradeep Kumar Das. Pradeep already did a presentation today, and now it's the second one. The topic is the perspective of manufacturer for the mouse weight gain test for mucil pertussis vaccines. So I'm very interested what will be in the presentation of Pradeep. Please, please start.
Yeah, thank you. Thank you, everyone. Let me share my screen. Can you able to see my screen?
Yes,
yes,
perfect.
Yeah. Thank you, everyone, for giving me once again the opportunity on the manufacturer's perspective for the mouse weight gain test for whole cell containing combination vaccines. So as mentioned by Catherine and my other colleagues, that there are some in vitro tests available, but most of the times what happens is due to constraints and due to other pharmacopoeia or regulatory requirements, we still follow the age-old techniques, which is one of the test is mouse weight gain test. So I will just wants to refer that as per the WHO TRS 941, where they have mentioned that manufacturers are encouraged to develop refinements and alternatives to mouse weight gain test. And in the next page, they have mentioned that a Chinese CHO-cell assay based on the clustering of cells after treatment with pertussis toxin is used in some countries to measure pertussis toxin in vaccine. A pertussis toxin standard is also included the assay, and the vaccine reference is used as a positive control. All the samples are serially dilated, obtained an endpoint, and the concentration of pertussis toxin of the test sample is calculated in relation to toxin reference. So the detailed procedure is also mentioned in WHO IVB 11.11, page number 190 to 197, where they have principally defined how this assay works. So basically, this assay is one of the most which we already did for the diphtheria, and now is the case where then the pertussis we would like to use this assay for routine batch release as per the pharmacopoeia. But as most of the pharmacopoeia does not have this CHO cell assay as an alternative, so it is very difficult for the manufacturers to universally allow and use this assay as routine batch release. So this assay has a assay validity criteria. That means no clustering in the negative control wells, clustering in positive vaccine wells, which is very few dilutions, and the difference between the duplicates should be less than twofold. And pass criteria has been defined, which should be 105 IU active PT per 0.5 ml of vaccine. So this is again, I have put a dimension how to do head-to-head comparison, where this is specific toxicity in vivo. You take 10 animals per sample and 10 for the control observation for 7 days, which is 0 day, 3rd day, and 7th day. Test is valid if no control group animal shows no weight loss or no deaths in any point of time. And your range should be meeting, the weight gain should be between 60 to 150%. So this is a standard things, and we know the pertussis is very labile component, and it has a lot of research is also required when you are doing this assay. It cannot be tested in any of the source of the animals. Manufacturer has to establish, confine the methodology, and the animal husbandry practice, means source of the animals, is most important and pivotal role in doing this assay. Otherwise, everywhere this assay is so variable, it cannot just give a head-to-head comparison where it can be tested in one site, it may give replicate, the other site will the same. So the source of the animal standardization is very complicated. In pertain to this, there is other method where the reference standards, everything is available, and you can develop this assay as an alternative. So this is a pertussis toxin detection, which is in vitro. Observation is only for 48 hours. Value should be less than 1 IU per SSD, and it is a fail and pass. So here in the cell control, which gives you no clusters, and clusters with the reference protein, you can find these clumps after 48 hours. And this is related to the cytotoxicity where the adjuvant, because your product contains adjuvant, it will have cytotoxicity, but once you serially dilute, the dilution factor, you need to take it into account. And then after 48 hours, when the vaccine you are calculating, it should be totally negative. So this is the very simplistic procedure, and results can be obtained within 48 hours. So this is one of the design of experiment. Here I have mentioned that this is the negative control where you are repeating all the neat sample. Here you the reference toxins, which is provided by the NIBSC, and you serially dilute. This is up to 4096. You put your sample 1 and 2 in duplicate, and again in the next well sample 2 in duplicate, and you serially dilute it. This dilution factor you have to optimize based on the cytotoxicity of your adjuvant component. So in the right side of the picture, you can see the negative means it does not have any cytotoxicity, and the reference toxin, it gives a 8 point. At the 8 point is 256 is your positive, and after 256, 5 and 2, it shows as a negative. So it shows very consistent approach when you are doing this sample. So when you are using vaccine, adjuvanted vaccine, it first gives your cytotoxicity, and then it comes as a negative. So this is the procedures you need to optimize when you are doing with a in vitro technique. So just I have given a dilutions, how you are preferring the dilutions. When you are taking the NIBSC reference standard, dilute the reference standard to 10 IU per ml, and you go serially. And this is replicate 1 and replicate 2, where you perform this assay, and then you calculate the geometric mean endpoint. Similarly, for your batch 1 and batch 2, you are getting cytotoxicities, and followed by when you calculate the endpoint dilution, at what dilutions you are getting positivity. So suppose it is coming as a 2560. So endpoint is 2560. When you divide with the dilution factors, like sample endpoint fold divided by the toxin endpoint fold into the reference concentration 10 IU per ml is the IU per SHD you can get. So here I get 50 IU per SHD. Similarly, here I got, for this batch, I got 25 IU per SHD. It is very, very simple. Initially, the standardization is a little bit cumbersome, but once you standardize, this method is rock solid. I will show one of the case studies also. So these are the one case studies which I have taken almost a 200 batches data from routine testing, and this green line indicates the CHO cell value, whereas the in vivo test indicates the blue color line. You see the similarities. Most of the things, it is coming less than 105 per IU per SHD. And it is very, very simple and robust, and it does not require any in vivo methodology, and results can be obtained within 48 hours. And it is very, very, as CHO cells are very, very sensitive, it is very sensitive to pertussis toxin. This can be one of the most alternative methods which we should try as a manufacturers with in compliance with the WHO guidelines for routine batch release. So this is the one case studies I want to emphasize to all of the team members, which is the manufacturing perspective to replace mouse weight gain test with a CHO cell assay. So I think this is my last slide for the conclusion. As we are working for the in vitro methods, which is much more reliable and validated, and which reduces the time for the batch release. Whenever there is an in vitro or alternative test is not available, we propose, based on the consistency approach and NRA approval, in vivo test may be omitted for routine batch release. And there is also a need, as I mentioned in this case studies, there is a mutual research work required for both industries and regulatory agencies for development and validated for robust 3R methods in a global scenario. Similarly, and the prevailing challenge for implementation of 3Rs, which currently lacks the comprehensive harmonization of regulatory requirements, it gives a little bit hindrance. So some of the, when we as a manufacturer export this product to other countries, they follow exactly what is mimicked for the WHO TRS or their own pharmacopoeia, where we as a manufacturer may have to do once again the test with the same animal model and submit the results. So this is the main concern for us as a manufacturers. If we can harmonize and we can replace these traditional methods, and it will be very useful for the time-bound manner to use alternative methods which are much more robust and valuable in reproducibility and precision. So that's all from my side related to CHO cell assay. Thank you very much.
Thank you, Pradeep. We have some comments in the chat. Since we have you here, if you still have a few minutes for us. So there are some comments about the fact that the mouse weight gain test is not only for the pertussis toxin. I don't know if you want to comment on that.
Currently, for the pertussis is the only mouse weight that is the approved method in pharmacopoeia for batch release. As I mentioned in my WHO guidance, they given a statement that manufacturers are encouraged to develop as an alternative method, but it requires a global scenario. Yes, this method can be used as an alternative to mouse weight gain test. So I mentioned that I have used around 200 batches data head-to-head comparison. So this data only will be applicable when I have a regulatory approval for doing this assay for routine batch release. Until then, I have to depend upon the in vivo methodology. So the chapter like 5.2.14 and in Indian pharmacopoeia also, they have given additional relaxation to remove from in vivo to in vitro. These strategies, at least in Indian scenario and wherever the WHO approved countries, we can utilize this methodology as an alternative.
Thank you, Pradeep. One question from me, and then one additional one from the chat, if we may. Have you already inquired with your national regulatory agency about the possibility to transition from the mice weight gain test to the CHO cells assays?
I have tried, Laura, but the only thing is, as in Europe, they don't use wholesale pertussis. So that is the main change. In European pharmacopoeia, it still says mouse weight gain test assay. But I would suggest if somebody can take up this matter for DTP chapter in European pharmacopoeia and provide an alternative methodology that will create a lot of help for other manufacturers to develop this method, and this can be also taken in certain context in our other regulatory agencies also. So this is only one DTP chapter which still says mouse weight gain test need to be performed.
Yeah, in the European pharmacopoeia, yes. You flag. Thank you for flagging that. And then the other question in the chat is about whether you can explain what exactly the clustering of the CHO cell is indicating. Is it just reflecting cell growth, so the column formation, or something else? And also, what is the cutoff for a cluster, for example, the number of cells?
So cell clustering is basically when the toxin is there, it will form a clustering of the cells. So there is a requirement given in WHO IVB. The clustering should be less than 10. Then it is you are considering as a positive. So you have to calculate based on your serial dilution at the end point where you are observing the clustering. So initial dilutions as the vaccine is adjuvanted, like aluminum phosphate or aluminum hydroxide, it will give you a cytotoxicity, complete cytotoxicity, which I mentioned in my slide as a C. You need to go for the dilutions, and then you have to consider the dilution factor, what you have used to greater the obtained should be less than 105 IU per SST per 0.5. So that is the main reason that the CHO cell clustering assay is more sensitive rather than any or mouse weight gain test. And mouse weight gain test looks like very simple, but when you are standardizing from starting from the strain of the mice, from husbandry to implementation of this mice, it is very, very tedious. You cannot use any single source of mice for use for this particular assay.
Thank you, Pradeep. And again, Juan Arstiniega from the chat says that the test was also used to detect the endotoxin or dermonecrotic toxin as well. So yes. Thank you, Juan, for your input on that. Okay, thank you, Pradeep, for being available for these questions. I hope that you can still stay online. Now we actually come to the closure of our presentations. And myself, together with Marie Schindel from AGES Austria, we are actually going to moderate a discussion session with all the speakers and also some of the members of the scientific committee that help us to put together this workshop. So I would like to welcome all the speakers and the members of the scientific committee that are online to turn on their video, and so we can start discussing together about some of the topics that came out within the chat and the Slido, but I think that we are here to really have a general discussion. And if I may just really say a few words, because so far what we have been seeing from the presentation is that we are in a very different landscape in terms of the possibility to transition from in vivo to in vitro for the safety testing of those products. So we have been seeing that a lot of effort has been put in order to develop new methodology to remove obsolete testing, but there is still a lot of work to do in some cases, just really to make sure that some of those opportunities can really be concretely used and implemented in a context that is the one of the quality control of those products. And at the same time, there is still the need to push for regulatory alignments and making sure that all the players and the stakeholders can really concretely consider this type of transition. So I think that's one of the interesting questions that actually came out through the Slido is, I think, very interesting to maybe open this discussion. It's again about the acceptance of those methodologies within the low and middle income countries. And we also started to have some discussions so far. But I would like actually to welcome any comments from the speakers and the scientific committee that are online.
So Laura, comments from whom you want actually? I think Sajad is joining.
Yes. So Pradeep, you already mentioned that you actually had some complexities to get your data accepted for the mice weight gain test. But if you can just share some thoughts from your perspective about what kind of complexity as a lower middle income country manufacturer you are experiencing when it comes to the acceptance of some of the methodologies. If you want to share what your company so far was able to implement and get accepted.
Once we see, basically we follow WHO guidelines on this for develop a method. First of all, it should be have a option to be available to do the analysis, because as a manufacturers, we need to develop the method, validate it, and then it needs to be submitted in a global forum. But the most challenging part will there once we develop the method. Most of the NRAs or most of the countries outside this one, they won't accept the alternative method until unless it is written very clearly in the guidelines. Yes, as an alternative, just like virocell assay, which says that for the diphtheria component, you can use either in vivo or in vitro method for routine batch release. Until that statement does not come in some of the pharmacopoeia, it is very, very difficult rational for the manufacturer to go and support on this. Maybe that NRA to NRA perspectives also it sometimes differ. So sometimes we get some of the observations from the NRA, which states that this test is not available in their pharmacopoeia. You have to provide an additional test on this. So we may have to perform the additionally test on this. So that's what we are pushing as an harmonized procedure wherever it is applicable in a global forum, that this test as an alternative test which is available should be available in the guidance document. It is very easy for us as a manufacturer to directly implement from in vivo to in vitro. As a manufacturer, I can say in my company is promoting a lot on the 3 Rs wherever, and all the supports are given to us to implement. But it should be under the regulatory framework. Thank you.
Does someone want to add here something, their perspectives to implement the in vitro methods? How they get in contact with the regulators?
Question 2.
Sajan, perhaps is it something that you can share from your perspective since you have been also.
Sorry,
Thank
could you
you.
repeat, because it was very hard to understand.
It's oh,
Okay.
sorry. Marie, do you want to please?
You can hear me?
Yes.
Is it okay? Okay. Could you please tell the audience your experiences how it is to get into contact with the regulators if you would love to implement an in vitro assay? Whom do you contact? What are your steps
Sure.
to succeed in this
Yes,
project?
indeed. Well, I would say if we take the example of the Binacle assay, so it was developed by the German Health Authority, so the PI. And clearly that was our first point of contact to understand the motivation, how far this would go. Now we are still in the development phase for it. However, then once we are convinced that this method can be really applied in our GMP setting, I think it would be really crucial to reach out to key regulatory bodies. Our tetanus vaccine is, I don't know the exact number, but it's between 150 and 180 countries marketed. So we will not be able to ask each of those regulators individually. So instead, we would go to 2 to 3 consultations. So in Europe, this is called a scientific advice. In the US, it is a type C or type D. And I think that is quite important. But it's also always quite a challenge because if we now go ahead, we validate the method, put everything in and say, hey, that's what we want to do. Do you agree? It's almost like if you submit. If they do not agree, then you have to redo the work. If you go in a much earlier development stage, then there's always the risk to get the feedback provided to say, on principle, we do agree, however, provide more details. And then you already go down the path. So I think that's always the challenge in those replacement projects, that in fact, you need to find the right time point to reach out, not too early, however, also not too late if there's a misalignment or different positions on elements. And I think what is quite important is, before massive animal studies are conducted, it's really helpful to understand what is the expectation regarding comparability, because you can hopefully reduce the number of animal tests you do, but also the animal testing takes quite long. For safety assays, they perform quite well because the readout is simple. But I think tomorrow you will discuss potency assays, and here the picture is exactly the opposite. So those assays have variants that you would not accept for any other quality assays, only for those.
Thank you very much, Shahan, for this insights. Yeah. We can speak of a European regulator, or ICTP in Austria. So the industry partners, the manufacturers, they come to us at the beginning with a national scientific advice. Of course, they, as I said, they have to find the right time point. So when I want to go to the regulator, and then the second step is then we always advise, and now you go to EMA scientific advice. So your local regulatory agency also can give you lots of advices what you can even follow up. So this is something which is given in Europe.
Mm-hmm. And I think there may be pathways in Europe anyway. I know the 3R, there's the EMA 3Rs working party that's very interested in alternative methods. In the past, they had the possibility to have a sort of grouped overarching advice that may be interesting to see if this still exists in some format. And if there is a group of like-minded manufacturers working on the same sorts of assays for implementation, to get an overview of what the feedback from this group is and how to go forward. And it will save maybe some individual discussions. It's, of course, not a regulatory decision, but it would be potential advice.
Thank you, Catherine. Indeed, that's something that's when within the projects that we are running, we have this type of conversation between among manufacturers versus the regulatory authorities. This is not something that is yet happening with the same type of like minds that it can happen within Europe, because I think that there is still some complexity in terms of having those methodologies be well understood by some of the representative, the national regulatory agencies or national control laboratories. So the fact that there is still a little bit of lack of knowledge or experience or direct experience on those, it's making this type of discussions a little bit more complex, because again, they rely on the fact that manufacturers are probably going to be the ones sharing in advance the data and their experience, but on the other side, they might not have any direct experience on those. And this is actually getting some not actually easy way to say, okay, shall we be the first ones to go and share? And this type of situation is bringing me to another topic that I hope that you will be also interested in commenting on, the fact that sometimes we have been seeing in the past 10 years while the power of working together through collaborative studies or making sure that there are small consortia or a joint assessment of those methodologies, because then there is definitely a different type of perspective if in approaching the regulators or engaging since the beginning of an assessment of a methodology, the national control laboratories that eventually will be also using the same methodology. But of course, we are in a situation where we can probably not have so many activities and projects in that directions. And this is probably where it's again becoming complicated, and we probably still need to rely on the fact that some of the manufacturers will need to take the lead in this type of strategic approach. So I don't know if you would like to comment. So many of you, they have a lot of experience in joining this type of projects or joint projects, but at the same time, many of you also have direct experience in going alone to the regulators. I don't know if you would like to share your experience. We heard about some collaborative study today, so if you would like to comment on that.
Yes, so maybe I can start if you like. And I will just stay on the Binacle, right? Because that was the presentation of today. There's quite a long history of different collaborations, and it's always a quite long way because it's more difficult to find a consensus. This requires more time than if a small group or organization goes alone. I think finally it provides quite a lot of value, because the conclusion to have a standardization is then always linked to the availability of what is required to replace the test. And this means at the end of the day, for the laboratory to have access to the critical reagents, but also to critical knowledge. So what are the key elements to look out to? Because the moment the development project stops, it switches over into life cycle. There's always the risk that you have the brain drain, because people will move on, they go to other activities. And then in 6-7 years, you face an issue and how to overcome this. And I think therefore the studies to really lay a great foundation to do so. The challenge, of course, if I now make the transition back to the Binacle, is yes, I think a really fruitful, promising method has been developed. However, clearly, if it is now handed over to different manufacturers, we will end up with different methods, with different sensitivities, because simply other reagents will be used. And I'm not an authority, so I'm not a decision maker to accept this. But if I imagine that the same person in an authority will get 2 submissions on the same topic, on the same test, coming with a factor 10, factor 30 difference on the critical quality attribute, the sensitivity, that will probably lead that one of the 2 will not get an approval. And then you will, in fact, at the end of the day, create a situation where you might not be able to replace the test, even so technically it's feasible. So therefore, I really think that these collaborations are really important. And they, however, require that the impacted industries are represented and are there and are sharing the critical elements.
Thank you, Sajan. Indeed, anyone else would like to comment? So, for example, now you brought up a very interesting case, because then we have the Binnacle that's actually reached out, something that we can say the full development is just a matter of hopefully solving the technical issues, but what if the Binnacle is not a perfect assay for your product? And that's where we have potentially the opportunity to see how the method that Andrew and his team is developing, but then what is going to happen if we will have two different approaches that are available, but maybe not fully accepted around the world, because again, for the lack of knowledge or understanding or access to the critical reagents. So because that's actually what we are going to see, and is going to be true for both the safety and the potency in this case.
Yeah, I mean, I think the two tests have their benefits and are not necessarily mutually exclusive. In cases, I think it will depend a lot on how QC friendly the two tests are. I think in regular production, if the Binnacle is relevant for your toxoid, it may in a QC setting be something that is worth investigating. The cell-based assay for those where it's not relevant, and for development, because this is also part of the vaccine life cycle, could be a way forward. But in both cases, I think, and not just for the Binnacle and for the toxicity, but for all assays that are developed as alternatives. And because there was a webinar on critical reagents a little while ago, also hosted by you, that it is a challenge. And I mean, we're talking about safety assays or safety tests right now. We're seeing more and more for the potency tests. There's no common reference material, so because it's very product specific. So I think there are ways to work under these conditions and still get products approved if you have a strong dossier that shows the method validation within your context for your product, with the references that you've chosen that are qualified and you've shown why they're relevant. Then I think this is not necessarily a blocking point. It just means that has to all be put in place. And with respect to the authorities having different opinions, I think this is unfortunately not a new thing. And that the more the authorities can have information on how the test works and invest the time in understanding the different assays and what the possibilities are, and really looking at them at the scientific level, and maybe have a few forums and spaces where they can interact themselves, either through different programs with exchanges between authorities that I know exists with the EMA. They have authorities that observe some of their procedures to see how it works and what kind of questions are asked. These are special relationships they set up. And to rely on the experienced authorities who may have seen these assays before and ask questions. This is also a way that we can move forward. But there are lots of new things coming. Everybody can't know everything, it's clear, but we can share our knowledge and make it better for us all.
Thank you, Catherine. While you are on that point, we have actually a question in the chat from Juan about what could be the role of DQM on the replacement project for products that are not actually based in Europe. And of course, the example of the wholesale pertussis came out really nicely. Pradeep mentioned that there is still one chapter that is mentioning the wholesale pertussis, but there is nobody in Europe that is producing wholesale pertussis anymore. So what do you think is going to be the role of DQM in supporting this type of activities, even if it's not your direct focus?
Well, I mean, I think your last words were the key point. It's they aren't our direct focus. So I mean, we, like everyone else, have a certain amount of resources, so our priorities are towards products that will be on the European market. And I think our doors aren't closed. Let's say if there was something particularly relevant and of interest, but our priorities are the products on the European market. And that's why you see we haven't had any progress on the wholesale pertussis side of things, because there are no wholesale pertussis products on our market. So I think there are other groups who still have this product on their market, and there may be ways for them to organize to bring this forward. Yeah.
Thank you, Catherine. And we will definitely get there on the webinar tomorrow on the potency side for the wholesale pertussis. So thank you. I would like to...
Sorry, just maybe I see a
yes.
comment from my colleague, Catherine, who wants to share that the tetanus monograph was just revised to include alternative methods for the absence of toxin test, and comments on the draft are very important. And this is also part of the global and international way to move forward and have everybody have confidence in the different alternatives that are put forward. So there's a link in the chat that you can use to comment on this text. Thanks.
Thank you, Catherine, for bringing it up. It's definitely important, and this is something that's I think that it's the role that we can all have, just to really monitor when there are opportunities to comment, not only within the European pharmacopoeia, but many other pharmacopoeias around the world. They are using the same system, so opening their chapters for comments. So it will be important, and I hope that through the project that we are doing, we try to share whenever there is the possibility to interact with the other pharmacopoeias and comment on that. And I would like actually to use this exactly this point to actually also call up for the two representatives from the WHO that are present today with us. So Dr. Ivana Netsevich and Dr. Jan Langley that are actually working on defining a guideline from the WHO on the transition from in vivo to in vitro, because that's the role of WHO here. It's also very important, and what you all have highlighted on the fact that there is important to have guidances and text, future-proof text, to allow those opportunities to be taken up also by other regulations. That's actually very important. And since there is a direct question in the slide about what could be the role of this guidance to promote harmonizations, I hope that Ivana or Dian Liang, they can comment on this.
Yeah, thank you, Laura. And incidentally, I'm working in WHO and responsible for development of international standard for vaccines and other biologicals. And the WHO, we strongly encourage manufacturers and regulators or other stakeholders to develop in vitro alternative assay method to replace animal testing. And Laura, you mentioned we are developing a new guideline for the name of the guideline has not been really decided, but it's kind of focusing on phasing out of animal test in the quality control for vaccines and biological products, either for lot release and for quality control, which will give a strong position from WHO point of view, and to introduce in vitro assay method to replace the animal testing. Including innocuity test, general safety test, potency, toxicity test, advantageous agent test, also Lao and parogenicity test, etc. So from WHO point of view, once alternative assay method had been developed and had been validated by the developer and also by stakeholders, WHO will be accept or recommend the method. The best situation will be the method had been approved or by a regulator and had a long experience and can be used for quality control of the product they are targeting. And that's how WHO TRS the presenters mentioned a couple of times, either it's in WHO TRS or in the manual we published the full quality control test on DTP, and where we include a lot of in vitro assay methods for the quality control of vaccines, especially for DTP. And once we heard a lot on the either it's a binacle or the cell-based assays for tetanus and botulism toxin. Once the method had been validated and been approved either by EDQM or by other regulators, and the WHO will put them in our TRS once we revise our current version of the document. Thank you very much.
Thank you, Dian Lang, for your perspective. I think that it's important, again, the role that you can have in order to, again, include those additional methodologies, again, to build more confidence on the fact that those can be accepted and be part of the routine strategy. So thank you for that. So something that I always communicate whenever I can is the fact that sometimes we cannot just wait for you. I think that it's also important that there is some knowledge sharing and experience sharing from companies or other regulatory authorities that are a little bit more far ahead in method validation and acceptance to really make sure that they share their experience also with others. This is actually very important, again, to make sure that there is more knowledge available, but also more trust on the possibility to transition to these new methodologies. And I think that if you agree, I would like to go back a little bit on the reagent piece, and thank you, Catherine, for bringing that up, because this is something that came out in both Sajan and Andrew's presentation about the importance of the availability of the critical critical reagents, because this is something that if we don't have that available, then the methodology cannot be used and cannot be validated or implemented. And there is a question on a slide about the use of the tetanus toxin, because this is definitely a key point, because it's probably not so straightforward to have a live toxin to be shipped around the world. So I don't know if Paul, you can jump in, because Paul Stickins from MHRA is the responsible of the reagents, and he is actually working to support many of you in WHO on that. So I don't know, Paul, if you would like to jump in and comment on that. Paul, can you try to speak again?
Yeah.
Okay, there is a lot of echo, so give. Let's see.
Stop. I might. But I think should be
Okay, Paul, I think that's, we cannot really hear you properly. Sorry.
I'm happy
Shalini,
to jump
yeah,
in.
please,
Yeah,
thank
Laura,
you.
if that's
Sorry,
helpful.
Paul.
Yeah, so there are challenges definitely to shipping tetanus toxin as we would propose in a reference reagent. And it might be that shipping costs would need to be a little bit more expensive in order to cover that. But in terms of stability, we would really be looking to get a really robust stable tetanus toxin reagent that we would robustly test before making it a reference reagent. So we hope that we'll be able to circumvent all those difficulties, and through the MHRA we should be able to easily ship worldwide through our standards department. So that's the plan, and I hope that we'll be able to overcome any kinds of issues that would come up.
Thank you, Shalini, because this is definitely something that we heard about, on the complexity that maybe are linked to specific legislation or a custom proceeding or procedures within many countries, and this has been flagged for many type of reagents or cell lines to be one of the big things to try to find a solution on. So I gladly the experience that you are having within MHRA might be able to overcome those, but at the same time we will need to again make sure that we are not actually going to get something similar to what is coming out within the Binnacle. That's maybe there are not suppliers that are interested to find a commercial way for some of those reagents. So hopefully whatever other methodologies that are currently in development or will be developed in the future, they won't have to deal with the same kind of complexity.
Should we move on? Question?
I think,
sorry.
No, I was going to say, in Shahjahan's presentation, I thought he had a nice idea. He talked about a consortium of companies, and maybe they can work via their associations or through some other platform to approach CROs with the appropriate GMP quality to show that there is a market for these products and that they could sell them if they make them. I think that's one of the challenges is either you have smaller organizations that have some resources but not a lot to supply the world supply for a particular reagent in the right quality, or CROs who don't want to launch into it because they don't know that there'll be a market for them afterwards. So is there any platform within the manufacturers associations that you could try and identify companies, show them that you would have that they would have a market for this and work some way to have better access to these reagents?
Yeah, it's a very good question, Catherine. I would say in Europe, yes, those organizations are definitely in place. If we think more globally, then I'm not sure if there is really a structure that, first of all, has the reach to all the manufacturers that could be impacted. Plus, they also need to provide the right legal structure, because even if no one will profit from this, make any money, it will still be considered a commercial activity, which will completely change the legal framework. And I think that I think we can definitely find a way forward in Europe and can try to be transparent and as open as we can. But to my knowledge, I do not know if there's really an organization available that could really cover this on a global scale.
And the DCVMN, would they have any ideas? Any members?
We just lost the CEO of DCVMN, who was with us till a couple of minutes ago. But I don't know if Pradeep, you would like to comment on that. DCVMN might be definitely.
Definitely. Yeah, Laura. DCVMN definitely will be part of any collaborative study, which as a manufacturers we can consolidate together. I know Dr. Suri sir is very much inclined on this front, where as a manufacturer, just like we did for the PSPT, similar way for any methodology, DCVMN definitely will be part of that initiative.
Thank you, Pradeep. I think that it will be interesting to see how maybe we can continue to have a conversation on some of those methodologies that are kind of available, and this is some of the conversation that we are having with the different companies from many of the DCVMN countries, really to try to understand exactly which of the methodology that you show today and presented today, and the ones that we'll present it tomorrow, they are going to prioritizing. So what is something that's which of them can be part of their short or medium term planning in terms of transition from in vivo to in vitro. And hopefully within this year, we will be able to share a little bit what are the trends or the ones that have been prioritized by the majority of them. And hopefully the idea is to, again, try to see if there will be any type of collaboration opportunities that could be supported or promoted, again, not only from the concrete implementation of the methodology, but at the same time also to find out ways where maybe critical reagents or critical reagents or cell lines can become available, especially in the low and middle income countries, where sometimes some of those big suppliers might not be super inclined to open up to those markets. So we really had an interesting experience through the pertussis serological potency test consortium that we set up within DTwP a few years ago, and seeing exactly the complexity, even to get some of the reagents that are not critical reagents to be maybe shipped or purchased by some of the members. So, and again, also realizing that some of the reagents that were available were not working properly within the different assays. So there is also this type of complexity to be considered, that products are so specific or so diverse that even a difference in a reagent can make an impact on the assay itself. So we really need to be aware about all these type of little details and again securing support and knowledge sharing between the many, just to make sure that if someone has encountered some of those complexity that are linked to the methodology itself or by the reagents, there is the knowledge shared, and you can maybe get in touch with the people that had similar experience, and maybe be reassured that maybe just by changing a reagent, the assay is going to work again. I don't know if this is something that has been experienced in the other, I don't know if in the various BSPs or in the other international collaborative studies, but this is something it's also part of the development of the methodologies and their validation.
Yes, indeed, Laura.
Let's go back to our Slido. I think that there are still a few questions, but please just if you have additional questions, just use the chat or the Slido. There is one for you, Sajan. It's more related to the binocle. So a person is actually asking if you have titrated the toxin used in the binocle, also in the guinea pig, to get an idea of the sensitivity or detection limits in the guinea pigs.
Yeah, it's a fair question. No, we have not conducted this. We have some quite historical data that give us a general understanding. However, it does not represent our current manufacturing process. And we're in fact today planning such a study. And this brings me back to the comment I made before, because we were discussing a lot, should we first reach out to the health authorities before we conduct the study, or do it up front? And finally, we concluded if we can't provide something in vivo, probably from the authority perspective, it will be very challenging to provide us any guidance. So we aim to do this in the first half of this year. And then based on these results, it will also set a good bar for us to understand where do we need to end with our in vitro assay.
Sajan, the question then that I have is it something that we should consider as a standard also to define the limit of detection of an in vivo assay, because that's something that could be complicated. So, and is it really needed to do so? Because otherwise the authorities are probably going to have less confidence. And then we go back to the fact that, do you really want to compare the limit of detection of a guinea pig based assay versus an in vitro? Does it make sense? Because this is some of the questions that very often comes out about the comparison in vivo, in vitro. And I remember that also something similar came out with other assays, asking somehow to understand what are the specification of an in vivo assay that could not be compared whatsoever with the current in vivo. Sorry to bring that up, but those are topics that are coming up very often.
No, it's a good and fair point. And in fact, we're a bit late with our study in the planning, since we're waiting on the feedback from another project where it's a link to an existing in vivo assay, where it's also a safety assay, where it's quite clear what is the limit of detection. And the feedback we got provided there by a major authority really made it clear that it is a requirement to have a direct head-to-head comparison for those kind of safety assays. So that's kind of a lesson learned for us to say, if you want to move ahead with this, we can really expect this as a requirement, especially since the products would very likely end up in the same department of that authority. Yes. But overall, I agree with you that it's always a challenging point. And if you would imagine that tetanus would not be toxic to guinea pigs or any rodents, how would you ensure it then? Then you would have set a different gold standard. And I think that's overall the challenge, right? Because you started with the animal, now it's the gold standard, while we know scientifically most of the time it is not.
I think, I mean, part of the goal with the safety test is to show that the test you're using is at least as sensitive as the test that you've used before. But I would imagine that if there are authorities that are insisting on this kind of data, the approach to showing this can be rationalized in a way that the number of animals is reduced with respect to the prep work that you're doing, so that if unfortunately you have to go back to the animal test to do this, then the fewest animals used possible. And that kind of data would ideally be shared in a publication that could then be used by others.
Indeed, and I mean, here that's the important piece is it will, however, only represent the manufacturing process of GSK. And that could be then a limitation for others. Because we have not aligned on the strain. So we use kindly different bacteria of Clostridium tetanus. We do not source the same kind of media, or we might even use different media for it. We yield different steps. We have different inactivation and purification steps. So the other elements which are still present due to the manufacturing process can differ, or the concentration can differ, and that can therefore shift your result. And then, because this is not well studied, we also, in fact, do not know if there could be an impact on the strain of guinea pigs that you used, etc., etc. So for me, it's always, of course, our aim is to share this data. However, I would not consider this then to become, oh, this is the standard. Everyone needs to reach this, otherwise it was not a success, because it's really an isolated case, very specific to this procedure.
I mean, I think maybe just to add into this, I mean, this is something we've seen. So I mean, if you even look at the historic literature, like there's a tenfold difference in what the sort of, the sensitivity limit of a guinea pig is. And actually using the cell-based assay, we're starting to test lots of different batches of tetanus toxin, commercial, and it's very, very clear, like the units are very different between them. And this is a challenge which the botulinum field has massively struggled with. So, if you look at Botox versus Dysport, the number of units on their label is completely different, even though, so there isn't a standard. And so I think this is one of the things that we really need to generate some form of standard which we can benchmark all the assays against, whether that's the binnacle or the cell-based assay. And, and how do we do that? And, I think that is a challenge. And so, whether we just, we have to take one batch of sample and we'll say, well, these are the units that we're going to get in our assay. I don't know. I mean, it is a problem though. And it's certainly not been, in the botulinum field, that's a market of billions of dollars and they've still not solved it.
I was wondering, so that there presently no tetanus toxin reference, but there is tetanus antitoxin. Is there not some way to show whichever toxin you're using, where it sits in relation to the antitoxin, and that gives you at least some
no, we
framework.
do. So we can use the antitoxin, but I mean, I think one of the challenges, is the specific activity of the toxins is potentially different. So you might have the same amount of toxins. So you'll have, I don't know, your 1 nanogram per mil, and actually the activity of that toxin might not be the same, and that could be due to strain differences or some of the toxins being inactivated. And I guess the thing for the toxoid manufacturers, a lot of them are not actually testing, that's not what they're after. They're not specifically testing how good their toxin is. It's obviously it's just, they inactivate it to make the toxoid. And so it is quite challenging, actually. So this is something we're sort of battling with a bit at the moment. It's like, how do we set, what is the standard for our assay?
I think this is
an
important.
Oh, sorry, go ahead, Catherine. Sorry.
No, I think it's an important challenge for these methods that are being developed in a state-of-the-art, scientifically sound way. But you have to keep in mind as well that you've been running an assay for years and years and years that has no reference standard at all.
Yeah.
Well, yes and no, because in the past we had the L+ test where you were identifying if your toxin is really toxic. However, I think today technologies where it's much clearer that when you blast the genomes and the proteomics even on street strains and on vaccine strains, you see on the elements that are important for toxicity, they're really extremely highly conserved. You have the same case as for botulinum, because it seems to be required to be toxic. That's why it's a bit like, yes, indeed, there's no reference. However, this is a compendial method, so it was not developed by industry. It was designed by the regulators, and I don't think it is required.
Not sure.
Because if you would add it, what would we see in addition, except that we would have more animal numbers in the assays finally. The other element I wanted to say on the tetanus toxin standard is that the one thing, of course, that we want to avoid is that on a long run, this kind of standard has to be calibrated in the animal test. So the target then, of course, needs to be, okay, it will be a one-time activity to show a comparison. But from that point forward, you would then only calibrate it back into your in vitro assay and do not fall back on the guinea pig assay.
I think that's an important point.
I think certainly in the botulinum field, Paul's trying to speak. I think in the botulinum field, that is a real challenge when they're constantly having to recalibrate their assays back against the animals. And it's like a requirement. And so you can see in their animal uses for the botulinum toxin manufacturers, they're constantly, re-requesting animals to recalibrate the internal standard. So it is definitely it's going to be challenging.
But it was achieved for other assays, right? So if you look on the acellular pertussis safety assay, it was achieved. If you look on the diphtheria safety assay, this was achieved as well. So I think it's a realistic target. I think the challenge here with tetanus is that the guinea pig is really very sensitive towards the toxin. So the bar is quite high.
So, and what about actually getting out from the concept of the reference and starting to use the in-house references? Again, to avoid to fall back to the need to recalibrate or to need to create completely new reference standards that are not calibrating with an in vivo test, but with an in vitro, because that's something that's came out, for example, with our experience on the serology for the whole cell pertussis. We saw that whenever we were introducing the international reference standard, all the values of the assay were actually going very bad. So all the curves were not actually calibrated correctly. So we were actually wondering about the role of using international reference standards that were developed for fully in vivo methodologies. Again, it's just for the discussion. I guess that here we will probably need to be very flexible on what the future is going to look like.
I think the advantage of an international reference toxin for that particular safety assay is that it will allow each manufacturer to show that their in-house assay shows the right sensitivity and makes it easier for regulators to approve it, because it kind of sets a comparison. And then besides that, what will happen in practical is that the continuous verification of your in vitro assay will be anyhow done with an in-house reference, because there cannot be enough international reference material available. So this will only be used much more periodically, I don't know, every 1 or 2 years, depending on your quality requirements, to say, okay, I calibrate my in-house against the international. Yes, okay, it's still matching, and then you continue testing with your in-house. If you do not have this international reference, it would mean each manufacturer has to develop 2 in-house reference assays exactly for that purpose.
Thank you, Sajan. Please, Heidemarie.
It would be worth shutting down. If you don't get this international reference standards, then installing 2 in-house reference standards.
Yeah,
At least you set up your method. So, and regulators, they are satisfied with international reference standards and also with the reference standards of the manufacturer. At least there has to be, which you can compare. So I was wondering, so if already the manufacturer's reference standard is used in your quality control, so why couldn't you also use this manufacturer's reference standard also for the alternative methods? So at least you have them one basis you can compare.
So I'm not sure if I got it right. So because today we do not have an in-house reference standard for tetanus toxin, so we would need to identify one first of all.
Yeah, this was one question I wanted to ask before. MHRA, what is the timeline to get such a tetanus toxin reference? Because to certify one is also need some time, so that we know each time when the manufacturers have to calculate until they receive it.
Yes,
Shalini.
So we're working on a research project at the moment, and it's in its relatively early stages, but we hope that we will have data by the end of this year, and that we'll know something about the formulation makeup for those tetanus toxins, and then from that we'll be able to know what we could supply potentially and how suitable it is. We really want to test it really robustly before we obviously produce it and put it out there for purchase. So,
and would you take I know there are 2 different approaches for international standards, and there's the reference reagent approach where you provide a common material that's used so that everybody's working with the same and it's been qualified, and then later it might take on an international standard status, but for the beginning it's helpful and it can happen a little bit more quickly because it doesn't have the same sort of qualification process. Is that something you're thinking about?
Exactly, yeah. So we're starting with a reference reagent that would be a non-WHO standard, and then we'll know from that whether we could make it into an international standard and make it even more widely available. And because it's so understudied in terms of stability of tetanus toxin, we really need to get that data before we can launch out into a full international standard. So.
And if I Shalini, that's really great to hear, and I think what would be extremely helpful if it's lyophilized, but I guess you're aware of this, because
yes, yeah,
will make it much easier, because the moment you have a standard, it's also a commitment, because it will mean one day it has been used up and then there's a need for a second one.
Exactly. So it's everything from shelf life to specific formulation to repeatability, whether that's in the binnacle assay or in the cell-based assay, and obviously to have some in vivo unitage that we can then trace back to link it up to previous work.
Thank you, Shalini. Just a second that I'm monitoring. There is a question from Slido that's about that is from Juan again. Thank you, Juan. Has comparison for pertussis toxin, the standard, the WHO and the DQM are labeled both ways for the animal and for the in vitro test. So do you have any comments on that? So yeah, I think that this is the current situation of many standards, probably, right?
I'm sorry, I didn't catch the question at all.
Oh, sorry, Catherine, it was a comment from Juan saying that's the current standard, that is the current standards from WHO and the DQM are labeled in both ways, so they can be used for animal and in vitro test.
I think that's very specific to the different reference preparation. I'm not sure which one he's referring to, but I think that there are cases where that does happen because the tests have different outputs and show up in the different scale. So this is something that we do see, but I'm not sure which one he's speaking of specifically. Maybe one of the NIBSC colleagues, or sorry, MHRA colleagues has some thoughts.
Yeah, absolutely. I think unitage is a real tricky thing with tetanus toxin. So, for example, the diphtheria toxin that we sell, it's a reference reagent that's in LF units. You know, LD50 or PD50 are kind of the well-established manufacturer unitages that would be expected. And then you're also thinking about activity in, say, nanograms per mil, say, as what's used in the Binnacle study, so in terms of the spiking in order to detect tetanus toxin. So really, an ideal standard would be able to tick at least a few of these different boxes in order to be the most useful for manufacturers. So yeah, we need some harmonization on that side as well.
And Shalini, talking about this topic about harmonizing these things is something that's again can come up within an alignment agreement between WHO and the other regulatory authorities, and I suppose also with your help, since you are the ones that are also setting up those references, this is something that could be led somehow and or be put into a kind of priority list to make sure that those are things that are going to happen.
Absolutely. I think at the moment, this work is linked to an NC3Rs grant that we have with the University of Sheffield. But we very much, we very much want to make this available and really want to study this in detail so that we have that data to support the robustness of it. So I'm happy to talk to manufacturers, happy to hear their take on things as well. Yeah.
Thank you, Shalini, and thank you. Please, Catherine.
No, so can I ask for the tetanus toxin is still in the feasibility phase, but is this been proposed to ECBS already, or are you looking at it as an MHRA standard first?
I think we're looking at it as just a reference reagent standard first, and then we'd look to go to the WHO after that point.
Thank you, Shalini. Paul, I don't know if you would like, you still have something that you want to comment on, since we see that you are back online. Happy to hear your thoughts.
Can you hear me?
Yes, much better than before. Thank you.
Can you see me?
No.
No, I don't know what's going on. So I suspect pretty much everything I was going to say has been covered now. I'm sure Shalini's explained what we're trying to do. I think the key point is that this is not a reference in the sense that you would derive your activity against it. It's purely to benchmark sensitivity. That's all it would be used for. And I think we would need to do some in vivo characterization initially in order to understand sensitivity of those models against this material if we end up with a standardized material. But after that, it's really how does that compare for the different in vitro assays, be it clinical, be it cell-based. And going forward, even though we would hope to produce a batch large enough to last a considerable amount of time, and it will be lyophilized to hopefully assure stability for a long time, a replacement batch wouldn't necessarily need to go through all of that in vitro characterization again, I think. But it's, we'll think about that more as we work through the project. So, thanks, Shani, for explaining that.
Thank you, Paul. I don't know if there are any other comments, but I think that there is another topic that has been brought up within this slide. It's about the experience on the safety assessment of vaccine adjuvants. I know it's not really the topic of this, but maybe before we close, I don't know if someone has any feedback about this. If there are any in vitro methodologies to assess the adjuvants.
I'm not aware of safety because our focus is much more on the QC and licensed products. So I would imagine it's something that is looked at much more during clinical development for others. But I'm not aware of safety.
No, thank you, Paul. So it's a little bit borderline with our scope. So thank you. Okay, so I think that we cover almost everything for today. I would like to check if maybe there are some last comments, questions, or remarks from the attendees, but also from the speakers.
I would say maybe just this has been a nice opportunity for sharing, to have a little bit of an informal chat on a large scale. And I think that sharing information, sharing data, sharing points of view is important for the way forward. So it's been a good exercise, and thank you for hosting. Yeah.
Thank you, Catherine. Indeed, we really hope to be able to give as many support as possible or answer as many questions as possible, since this is a transformation that requires investment, time, knowledge, and there are a lot of aspects that might require expertise that might not be immediately available everywhere in the world. So hopefully this type of events, but any further guidelines and activities that are coming out in the next future will be providing additional support. And again, if someone has something else to say, happy to give the floor. Otherwise, I'm happy to transition you to the end of this meeting and introduce you the one for tomorrow. Okay, then.
Bye-bye. Thank you.
Thank you very much. So
Bye-bye.
I see you tomorrow for the potency workshop. So we hope to see you online, and thank you very much for being with us. Do not hesitate to write back to me if you have any further questions about the topic. If you need to learn more, presentations and recordings will be available shortly after this meeting on the event page. And again, do not forget to continue to follow the activities of the Animal Free Safety Assessment Collaboration. We do look forward to see you online and to join all our next events. So thank you very much again. Wish you a very nice rest of the day. Thank you very much to all the speakers and the scientific committee for their support in the organization of these events. Thank you.
Bye-bye.